Hello, everybody, and welcome to Mr. Saucedo's YouTube videos. Today, we're going to finish up talking about Mendel's experiments and his conclusions. So we're going to talk about how you can predict genetics now. So we did this all without really going into the specifics of genetics. We were just talking about what Mendel's experiments were, what his conclusions were, what his results were. That was it. So here's the thing about Mendel's experiments and why he's considered the father of genetics and not somebody else, even though he wasn't technically, a scientist by trade. So he performed his experiment thousands and thousands and thousands of times. And again, no one knew about this until after he died. It's kind of sad. But this is what he found, is that for doing that second experiment, where he used the F2 generation and he was looking at all these different traits, this is what he found. The dominant trait showed up this number of times. The recessive trait showed up this number of times. So let's calculate percentage-wise what that ends up being. Well, take a look. 25.26, 24.94, 24.11. What do we notice about these numbers? They're all about a quarter. Dominant, on the other hand, again, you can see there's some variety here, but all of them are about 3/4, about 75%. So what he observed is that in his F2 experiment, for regardless of what trait he was looking at, 3/4 of the pea plants always had the dominant trait and a fourth of them had the recessive trait. So Reginald Punnett came up with a way of predicting what Mendel saw. And this is what Reginald Punnett looked like. So here are our guidelines for how to kind of like utilize Punnett squares. Uppercase letters represent dominant alleles. Lowercase letters represent recessive alleles. And so, for example, blue eyes, that is recessive. So we would use a lowercase b to represent that. Whereas brown eyes would be dominant, and that would be a capital letter B instead. Now, keep in mind, a lot of times people get confused because they're like, oh yeah, blue eyes, brown eyes. They both start with the letter B. I'll use the letter B to represent them. That doesn't really matter. We only use one letter for each trait. So if we're looking at height, like let's say tall plants are dominant, I would use a capital T. Short plants are recessive, I would use the same letter, a lowercase t. I wouldn't all of a sudden change that to like a lowercase s. So we choose a letter to represent a trait, in this case height, and then the dominant one, we capitalize it. And the recessive one, we use the lowercase version of that letter. Okay. So we have different terminologies now that we need to remember, and these are really important. So first things first, homozygous, homo means same. That means you have two copies of the same allele. So you could have two uppercase letters or two lowercase letters. Heterozygous means you have two different alleles. So an uppercase letter and a lowercase letter. And so here we go. We have our parental generation where we had homozygous dominant parent. That would be the purple one. We had a homozygous recessive parent that would have two lowercase letters. Okay. And so, for example, two capital letters, like we just said, or a capital and a lowercase, like the F1 generation, or two lowercase letters, like the white flower here. Each one of these is a possibility. And again, I'm just using the letter G as a generic. It can be anything. It can be any trait we're looking at here. So these are the same. So that's definitely homozygous. These are different. That's heterozygous. These are the same. That's homozygous. Now, how do we distinguish between capital and lowercase letters? Well, we add the word dominant or recessive. So homozygous dominant is two capital letters. Homozygous recessive is two lowercase letters, right? So if I'm going through this, I should be able to kind of label this with which one is which. So this is homozygous dominant. This is homozygous recessive. This is heterozygous. This is homozygous dominant. These are both heterozygous, and this is homozygous recessive. All right. Now, genotype and phenotype. Genotype is your genetic makeup. So when you think geno, think genetic. Those are the actual alleles. Homozygous dominant versus heterozygous versus homozygous recessive. That is your genotype. Your phenotype, though, is your physical characteristic that you see. Blonde hair, brown hair, etc. So notice that because of Mendel's original conclusion about the principle of dominance, all it takes is one capital letter and you immediately have or show the trait for that dominant trait. So, for example, there's only one way to have blue eyes, according to this Punnett square here, right? And so that's if you have two lowercase letters. But there are two ways for you to have brown eyes. You could have two capital letter B's or a capital letter B and a lowercase letter b. Why? Well, remember, the capital letter always overcomes the lowercase letter. So if you're heterozygous, you show the feature of the dominant trait. If you're homozygous dominant, of course you're going to show the dominant trait, right? There's nothing else for you to do. So we got to keep that in mind when we're doing our Punnett squares. So how do you construct a Punnett square? Well, to make a simple monohybrid is what this is called, Punnett square, you construct a square with four windows. So I can just draw some lines in, four windows. You write one parent's genotype on the top, and we separate the alleles. We write the other parent's genotype on the left side, and we separate the alleles. So I'm going to use the letter Q, because Q can be anything. So let's make this parent homozygous dominant. Let's make this parent heterozygous. So we have an uppercase letter and a lowercase letter Q. Then we cross them. So here I would fill in Q's going down. Here I would fill in Q's going down, and then I go across. So I would have Q's going across. I would have Q's going across. So what do we notice about our offspring? These are our four hypothetical offspring. 50% of them are homozygous dominant. 50% of them are heterozygous. Okay, great. So this is genotype information here. Okay. We also want to know the phenotype, though. What feature are they going to show, though, all four of these? Well, 100% of them are going to show the dominant trait. So for phenotype, because the genotype is here in the boxes, 50% homozygous dominant, 50% heterozygous, I like to color in. And so I would choose, for homozygous dominant, in this case, it would be red, because that's the color of the Q here, the capital letter Q. If it showed a recessive trait, I would show it as blue. But you can see all of these offspring are going to show the dominant trait. All right, so let's repeat this for Mendel's first experiment. We have P, capital P being purple, that's dominant. Lowercase b is recessive. So remember, both parents were homozygous because they were pure. And so we have two capital letter P's for homozygous dominant for one parent. The white flower, that would have two lowercase p's, that would be homozygous recessive. So we separate out the alleles. And now we fill them in. So fill in this chart, going down and then going across. What do we get? We get pure heterozygous offspring, all four of them. So if I said, what's the genotype of your four offspring here? 100% of them are heterozygous. What's the phenotype? 100% of them are purple. And that explains his exact experiment that we saw, right? That's exactly what we saw. It was the first generation, we only had purple flowers. But let's take it a step further and do the Punnett square for his second experiment. Again, capital letter P is purple, it's dominant. Lowercase p is recessive. He crossed the mixed offspring. So he crossed two heterozygous purple flowers, okay? Remember, separate out your alleles. And now this box, we would have this. These boxes, we would have this. And this box, we would have this. All right, let's do percentages. 25% would be homozygous dominant. 50% would be heterozygous. 25% would be recessive. And what did he see? He saw 3/4 of them were purple. 1/4 of them was white. This perfectly explains it, right? And notice he didn't know the genotypes. He didn't. He couldn't have known the genotypes. We didn't know about alleles or genetic information. But it just so happened that if he had figured out what the genotypes were somehow, about 1/4 of them would have been purely like the P generation's purple flower parent. Sorry, did I say 50? I meant 25%. 25% or 1/4 would look exactly like the P generation white flower, homozygous recessive. And then 50% of them would look like the current generation's parents, the F1 generation's parents. They would be mixed. And that is how we, construct Punnett squares. So if you have any questions, let me know.